Prescribon Medical Education Series. Circulation of Blood, Part 1. Circulation of blood. The function of circulation is to serve the needs of the body tissues, to transport nutrients to the tissues, to transport waste products away, and in general to maintain an appropriate environment in all tissue fluids for optimal function of the cells. Blood flow is controlled mainly in response to their need for nutrients and removal of waste products and metabolism. In some organs, such as kidneys, the circulation serves additional functions. Blood flow to the kidneys, for example, is far in excess of its metabolic requirements and is related to its excretory function, which requires that a large volume of blood be filtered each minute. The heart and blood vessels, in turn, are controlled to provide cardiac output and the arterial pressure needed to supply adequate tissue blood flow. Characteristics of the circulation. The circulation of blood is shown in the figure, distribution of blood in various parts of circulation. In this figure, red color represents arterial blood and black color represents venous blood. The arrows show the direction of blood. Direction of blood flow. Venous blood looking less reddish, oxygen low, carbon dioxide laden, nutrient poor, and carrying waste products from all over the body are transported by the superior vena cava from upper parts of the body and the inferior vena cava from lower parts of the body and drain into the heart, first to right atrium, thence to right ventricle, from where it is pumped into the pulmonary artery to lungs, where exchange of gases, oxygen absorbed into the red blood corpuscles, carbon dioxide comes out of the blood and mixes with the air in lungs. All this take place. When the blood coming out of lungs, looking more reddish, well oxygenated, carbon dioxide poor, is transported by the pulmonary veins to the left atrium of the heart, thence to left ventricle, from where it is pumped out to the body through aorta and its branches all over the body. Systemic circulation from the heart to tissues through arteries and from tissues through veins back to heart is called systemic circulation. From the heart to lungs through pulmonary artery and back from the lungs to heart through pulmonary veins is called pulmonary circulation. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Now, getting back to circulation, the function of the arteries is to transport blood under high pressure to the tissues. The arteries have long, strong walls and the blood flows at a high velocity. The arterioles are the last small branches of the arterial system. They act as control conduits through which blood is released into the capillaries. Arterioles have strong muscular walls that can close the arterioles completely or by relaxing can dilate the vessels several fold. The arterioles can vastly alter blood flow in each tissue in response to its needs. The function of the capillaries is to exchange fluid, gases, nutrients, and other substances between the blood and tissue fluid, that is interstitial fluid. The capillary walls are thin enough for such exchange. They have minute capillary pores permeable to water and small molecules. The venules, small veins, collect blood from the capillaries and join together to form progressively larger veins. The veins function as conduits for transport of blood from the venules back to the heart. The veins also serve as a major reservoir of extra blood. The vein pressure is low and hence the walls are thin. Even so, they are muscular enough to contract or expand and serve as a reservoir for the extra blood, either a small amount or large amount depending on the needs of the circulation. Volumes of blood in different parts of the circulation. See the figure. Systemic circulation has a proportion of 84% of the blood being circulated. Arteries have 13%, arterioles and capillaries 7%, veins 64%, heart and lungs 16%, heart 7%, lungs, pulmonary vessels 9%. Note that only 7% of the total blood is in arterioles and capillaries, where the most important function of the circulation occurs, that is, diffusion of substances back and forth between the blood and tissues. Cross-sectional areas and velocities of blood flow. If all the systemic vessels of each type were put side by side, their approximate total cross-sectional areas for the average human would be as follows. Blood vessel and cross-sectional areas in centimeter square are shown. Aorta 2.5, small arteries 20, arterioles 40 square centimeter, capillaries 2500 square centimeter, venules 250, small veins 80 and vena cava 8 square centimeter. Note the 80 square centimeter area of the small veins as compared to 20 square centimeter for the small arteries, a fourfold increase in veins, explaining the large blood storage capacity of the veins as compared to the arterial system. Also note the enormous increase of the cross-sectional areas of the capillaries, that is 2500 square centimeter, as compared to arterioles 40 square centimeter. The blood volume F being the same in each minute, the velocity V is inversely proportional to the vascular cross-sectional area, that is velocity by blood volume by the cross-sectional area. V is equal to F by A. Thus, under resting conditions, the velocity averages about 33 cm per second in the aorta, but is only 1/1000th as rapid in the capillaries, about 0.3 mm per second. However, because the capillaries have a typical length of only 0.3 to 1 mm, the blood remains in the capillaries for only 1 to 3 seconds, which is surprising because all diffusion of nutrient food substances and electrolytes that occurs through the capillary walls must be performed in this short time. Pressures in the various portions of the circulation. Because the heart pumps blood continually into the aorta, the mean pressure in the aorta is high, averaging about 100 millimeters of mercury. Also because heart pumping is pulsatile, the arterial pressure normally alternates between an average systolic pressure level of 120 mm and a diastolic pressure level of 80 mm under resting conditions. As the blood flows through the systemic circulation, its mean pressure falls progressively to about 0 mm by the time it reaches the termination of the superior and inferior vena cavae, where they empty into the right atrium of the heart. The pressure in many of the systemic capillaries varies from 35 mm near the arteriolar ends to 10 mm near the venous ends, averaging 17 mm, low enough that the little of the plasma leaks through the minute pores of the capillary walls, even though nutrients can diffuse easily through these same pores to the outlying tissue cells. In some capillaries, such as the glomerular capillaries of the kidneys, the pressure is considerably higher, averaging about 60 mm and causing much higher rates of fluid filtration. In the pulmonary arteries, the pressure is pulsatile, just as in the aorta, but the pressure is far less. In the pulmonary artery, systolic pressure averages about 25 mm and diastolic pressure about 8 mm, with a mean pulmonary arterial pressure of only 16 mm. This means pulmonary capillary pressure averages only 7 mm. Yet, the total blood flow through the lungs each minute is the same as through the systemic circulation. The low pressures of the pulmonary system is good enough for the needs of the lungs because all that is required is to expose the blood in the pulmonary capillaries to oxygen and other gases in the pulmonary alveoli. Basic principles of circulatory function. Three basic principles underlie all functions of the circulatory system. 1. To each according to its need. Blood flow to most tissues is controlled according to the tissue needs. Active tissues need an increased supply of nutrients and therefore more blood flow than when at rest, occasionally as much as 20 to 30 times the resting level. However, the heart normally cannot increase its cardiac output more than 4 to 7 times higher than resting levels. Therefore, it is not possible simply to increase blood flow everywhere in the body when a particular tissue demands increased flow. Instead, the microvessels of each tissue, especially the arterioles, continuously monitor tissue needs such as the availability of oxygen and other nutrients and the accumulation of carbon dioxide and other tissue waste products. These microvessels in turn dilate or constrict to control local blood flow at the level required for the tissue activity. Also, nervous control of the circulation from the central nervous system and hormones provide additional help in controlling tissue blood flow. Cardiac output is the sum of all the local tissue flows. Heart automatically varies cardiac output according to tissue needs aided by nervous control. When increased volume of blood flows through a tissue according to increased tissue needs, it immediately returns by way of the veins to the heart. The heart responds automatically to this increased inflow of blood by pumping it immediately back into the arteries. Thus, as long as the heart is functioning normally, it acts as an automaton responding to the demands of the tissues. The heart, however, often needs help in the form of special nerve signals to make it pump the required amounts of blood flow. 3. Arterial pressure regulation is generally independent of either local blood flow control or cardiac output control. The circulatory system has an extensive protective system for controlling the arterial blood pressure. If at any time the pressure falls significantly below the normal level of about 100 millimeters, nervous reflexes bring about circulatory changes to raise the pressure back toward normal within seconds. The nervous signals forces the heart to pump more powerfully, contracts large venous reservoirs to provide more blood to the heart, and cause constriction of the arterioles to provide more blood in large arteries to increase the arterial pressure. Then, over more prolonged periods, hours and days, the kidneys play an additional major role in pressure control by secreting pressure controlling hormones and regulating blood volume. Blood flow. Blood flow rate means the quantity of blood that passes a given point in the circulation in a given period of time. Ordinarily, blood flow is expressed in milliliters per minute or liters per minute. The overall blood flow in the total circulation of an adult person at rest is about 5000 ml, that is 5 liters per minute. This is called the cardiac output because it is the amount of blood pumped into the aorta by the heart each minute. Laminar flow of blood in vessels. See figure flow of fluid or blood. When blood flows at a steady rate through a long, smooth blood vessel, it flows in streamlines with each layer of blood remaining in the same distance from the vessel wall. Also, the centralmost portion of the blood stays in the center of the vessel. This type of flow is called laminar flow or streamline flow, in contrast to turbulent flow, which is blood flowing in all directions in the vessel and continually mixing in the vessel, as discussed subsequently. Parabolic velocity profile during laminar flow. When laminar flow occurs, the velocity of flow in the center of the vessel is far greater than that toward the outer edges. In figure A, a vessel contains two fluids, the one at the left colored red and the one at the right a clear fluid, but there is no flow in the vessel. When the fluids are made to flow, a parabolic interface develops between them, as shown 1 second later in figure B. The portion of fluid adjacent to the vessel wall has hardly moved. The portion slightly away from the wall has moved a little distance, and the portion in the center of the vessel has moved a longer distance. This effect is called the parabolic profile for velocity of blood flow. The cause of the parabolic profile is as follows. The fluid molecules touching the wall move slowly because of adherence to the vessel wall. The next layer of molecules slips over these, the third layer over the second, the fourth layer over the third, and so forth. Therefore, the fluid in the middle of the vessel can move rapidly because many layers of slipping molecules exist between the middle of the vessel and the vessel wall. Thus, each layer toward the center flows progressively more rapidly than the outer layers. Turbulent flow of blood under some conditions. When the rate of blood flow becomes too great, when it passes by an obstruction in a vessel, when it makes a sharp turn, or when it passes over a rough surface, the flow may then become turbulent, or disorderly rather than streamlined. See Figure C. Turbulent flow means that the blood flows crosswise in the vessel and along the vessel, usually forming whirls in the blood called eddy currents. These eddy currents are similar to the whirlpools that can be seen in a rapidly flowing river at a point of obstruction. When eddy currents are present, the blood flows with much greater resistance than when the flow is streamlined because eddies add to the overall friction of flow in the vessel tremendously. The tendency for turbulent flow increases in direct proportion to the velocity of blood flow, the diameter of the blood vessel, and the density of the blood, and is inversely proportional to the viscosity of the blood. In large arteries, there is almost always some flow turbulence at the branches of these vessels. In the proximal portions of the aorta and pulmonary artery, during the ejection phase by the ventricles, there is considerable turbulence in the aorta and pulmonary artery, where many conditions are appropriate for turbulence, such as 1. High velocity of blood flow, 2. Pulsatile nature of the flow, 3. Sudden change in vessel diameter, and 4. Large vessel diameter. However, in small vessels, there is no turbulence. Now, dear listeners, this is the end of part 1 of circulation of blood. Part 2 is another video, Prescribon Medical Education Series, Circulation of Blood, Part 2.